Good day, everyone. Thank you for that introduction, even though. So today I am going to give you a presentation on the matters of the heart. And I don't mean that in a romantic sense. I mean that in a disease sense. So the title of my talk is Identifying Genetic Biomarkers of Dilated Cardiomyopathy using whole exome sequencing data from Southern African patients. So this is the work. I was approached by a group from Charlotte Maclege Hospital. So this is the division of cardiology, where they had identified patients who had a serious case of this dilated cardiomyopathy. So what they did is they recruited a number of patients where they looked at the left ventricle ejection fraction of less than 40%. Also looked at whether the source of the cardiology was of unknown origin, and then looked at the years, anyone who's at the age of over 18. And then they also excluded a number of patients who did not, who had all of the other disease mentioned in red, so as to not confound the results of the study. So they recruited in total about 101 patients and then did exome sequencing on those patients. And then they had FASTQ files. So they approached me with these FASTQ files and said, can you please help us identify variants that might be associated with the matters of the heart? So I'm not going to try and embarrass myself and try to explain what dilated cardiomyopathy is. So what I'm going to show you is these two pretty images of the heart. So on the left, on that side, that is a normal heart. And on the right here, this is a heart with dilated cardiomyopathy. So what you will notice is that the left ventricle of the heart is enlarged. And then the wall on the side is sort of thin. So there's a thinning of the heart muscle on the outside. And so patients who have this dilated cardiomyopathy will present with a wide variety of phenotype, from like your heart beating very fast to try and compensate for the blood not flowing directly into your heart. And a wide variety of phenotypes. Yeah. So what did we do then with the data? I'm going to try and go through this very slowly. If you do get confused, feel free to stop me and I will explain. So what we did on the left there at the beginning is we did variant calling. So we did variant calling using the traditional tool, GATK, following the best practices. And then we also wanted to compare it to the newer tool, deep variant. So we compared variant calling from deep variant and using GATK. And then after that, we annotated using VEP. So VEP is a variant effect predictor, which basically predicts what the variance, the phenotype is. And then after doing that, we sort of looked at only a set of genes that they were interested in that are associated with cardiomyopathy. So that was a list of about 14 genes. And then after we filtered the genes from hundreds of thousands, we ended up with a couple of thousands of genes that we took, and then we loaded them onto this tool called HALE. So HALE is a tool that is very useful when you are analyzing variants. It takes your VCF and puts it in a sort of a elegant data structure where you are able to analyze each variant according to the according to the qualities, the matrices, and everything that is associated with that variant. And then after loading it in there, then we started looking at what each variant was annotated with according to VEP. So the first filter that we looked at was impact. So any variant that was annotated with low impact, we got rid of. The next thing, we looked at consequence. In consequence, we were looking for anything that does not have intron variant in it. So we removed any variant that was associated with intron variant. The next thing, we looked at the allele frequencies for all of the variants that we had. So when we're looking at allele frequencies, we first looked at allele frequencies in African populations, looking at what each variant, what is its allele frequencies according to the databases annotated within African population. So we looked at NOMAD, and we also looked at 1,000 genomes. So we used an allele frequency of 0.5, which is 5%. The next, we look at the allele frequencies in our data set that was calculated during variant calling. And again, we applied a variant filter of 0.5%. So anything higher, we got rid. The next thing was the allele depth, the genotype quality, and then, so this is one of the filters that is sometimes overlooked. So when you're looking at allele depth, so you're looking at, say, for example, you have a genotype that is homozygous reference. But when you look at the reads that are mapped to the reference, you find that even though it's homozygous reference, but on the alternate allele, there's more than 10% of reads that map to that region. Obviously, there's something wrong going on there that the caller didn't do right, so you get rid of all those variants. Or if you have a homozygous, a heterozygous that does not have an allele balance of 1:1 or 50%, anything that is outside of that window or closer to 50%, you basically get rid of. And then you also filter on genotype quality and NDP. So anything with the genotype quality of less than 20, you get rid of. And then the next one, we looked at homozygous reference allele, and the homozygous reference allele we get rid of because it's not informative to us. And then we look at computational damaging scores. So we looked at things like CAD, your Revel, and so on. And we applied the recommended cutoff score for all of those computational damage scores. And then finally, after filtering all of those variants, we looked at how many variants were left without any sample assigned to it, or how many samples were left without any variants, and then we got rid of. And then in total, we ended with about 57 variants that were called from div variant, and about 41 that we call using GATK. So after doing that, we went on to do variant prioritization and classification. So here we look at what the ACMG guidelines say about the variant that we identify, and what are they annotated, whether they're pathogenic, non-pathogenic, and so on. So out of the variants that we find, we picked out about 16, which were annotated as variants of uncertain significance, and then about 6 that were pathogenic. We had like stop gains, missets, sparse junction variant. We had a stop gain again and the frame shift variant. So these are some of the variants that are highly associated, that we think are highly associated with dilated cardiomyopathy. Right? So what does this all mean? So we have sort of generated a workflow from going from raw data that you get from your sequencing. You align, you annotate, and then you filter, and then you're left with a certain number of genes that you can then take and to the clinicians, and then you do your sort of variant classification. So this workflow that I presented to you is reproducible. We've got containers, it's written in Nextflow. So basically, even if I run this next year, I'm going to get the same results. So the variants that we identify, they show a very strong association with DCM, but we're still doing further analysis and annotation to figure out whether are they really associated with the disease. And then finally, because when we were doing the annotation, a lot of variants that we picked up, they didn't have any any annotations with them, because the databases are mainly created on European and East Asian populations. But in our African populations, we know that our genome is very diverse. We've got lots of variants, and some of them do not exist in these databases. So this also raises sort of a gap where we think that we should try and prioritize in things like creating resources for variant annotation, and also looking at a similar guidelines for classifying a variants, but for an African setting. And with that, I would like to thank my supervisor, Scott, also Prof. Ngoba and Dineo, who brought this work to me, and also the collaborators from Vanderbilt University. Thank you.
Thank you very much, Phelelani, for keeping to time. Yes. I appreciate you. Let's take a few questions. Dr. Tobias.
So, very, very good talk. I have two quick questions. Question number one, what is the sequencing depth?
So the sequencing depth, if I'm not mistaken, was about 40 or 50
Okay,
X.
fantastic. So that means that you are able to pick up loss of heterozygosity and stuff like that. Now, your filtering was quite strict.
Yes.
Do you worry that you can miss things like uniparental disomy? Yeah, over there.
This thing is slow, sorry. So with our filtering, so like I said that a lot of variants didn't have any annotations associated with them. So when we are applying our filters, we made sure that we do not include and, but instead we use or. So we say that if for this filter you do find something, okay, it's cool, keep it. But if there's nothing there, it's fine, pick it up, and then we'll see what the other annotations are for that variant. So we were strict, but not that strict in a sense, but we did like leave room to say we know that there are some variants that are not going to have any annotations, but we still want to keep them and see what the final outcome is.
Phelelani, very good talk. I have a small question. Maybe I've got it wrong way. It looks for me that you consider each mutation as independent one from another. Did you try to do such epistatic study where you can see combinations of mutation? Some there may be compensatory mutation, you have bad mutation, but other hide it from disease. So it's maybe very interesting to find combinations.
No, definitely, that's something that we can consider, but we haven't looked at any epistasis.
Hi, good talk. I wanted to ask about GATK versus deep variant. So like deep variant identified more variants, it seems like, but like, I don't know, do they identify the same? Like, is the overlap, is the union the same, or is it like,
Yes.
are you identifying different variants?
So there was a huge overlap because you can see like the final, there's like a difference of, I think about 16 or 17 variants, but there was an overlap, but most of the variants that deep variant picked up were either of very low quality when we were doing the filtering. So that's how we got rid of them. Like I think when you look at, okay, I can't see it. But basically most of them were of very low quality and some didn't have any annotation. That's why they got lost.